on and just that black line. Okay, 2018 multiple choice, here we go. Diagram shows a cell with a section of the cell membrane magnified, so zoomed in section of what's going on and just that black line. Okay, molecule X is, just go straight know this, two parts of the plasma membrane that you have. You've got the phospholipids and you've got the proteins. These are the phospholipids. That is a protein. Okay, cellulose is not involved in this, that's part of the cell wall in plants, and starch is a storage carbohydrate. Okay, I hope you got to the 50-50 at least. Right, diagram shows an experiment in which a model cell was placed in a sucrose solution. So it's 5% on the outside, 15% on the inside. So that means that I've got a low water inside, high outside. It says at the start of the experiment, the model cell weighed 25 grams, and at the end it weighed 30 grams, which would not surprise you because you've got that high water, low water thing. What was the percentage increase in mass? Okay, percentage increase in mass, look for the same thing every time. I'm looking for the change, and then I'm going to divide it by the start, and then I'm going to times it by 100. Okay, the change was from 25 to 30, so I've got a change of 5. My start was 25. Okay, so all I'm looking for is to do that sum, times it by 100, and there we go. Or you could look at that and just go, that's a fifth, and that's 20%. Okay, question 3. Glucose molecules in low concentration of the kidney have to be moved into the bloodstream where there's a higher concentration of glucose. The process responsible for this action is, so I'm going from a low to a high. This is me basically trying to push it up the hill. Okay, if I'm trying to get up the hill, I'm going to have to put some energy in. It's going to be active transport. Okay, it's definitely not osmosis because that's water. Diffusion is is high to low, and passive transport would be no energy involved at all. Okay, question 4. Which of the following represents the sequence of events in the production of a protein? Okay, so we're going to end with protein. We need to start with a code. So I need to start at DNA. So get rid of C and D straight away. Okay, so DNA copies a message into mRNA, and then that mRNA codes for the amino acids, which finally give you protein. You just have to know that. Okay, which of the following are all types of proteins? Okay, so hormones, yes. Enzymes, yes. Nitrates, no. Okay, they are used to make proteins in nitrogen cycle, but they're not a protein themselves. Antibodies, yes. Enzymes, yes. Plasmids, no. They're made of DNA. Hormones, yes. Receptors, yes. And antibodies, yes. So we like C. Just to be clear, receptors are good, antibodies are good, nitrates we've already said are wrong. So definitely C. Question 6, we've got stages in genetic engineering. So we've got identify required gene from source chromosome, extract it. And then on the right hand side we've got extract the plasmid, stage X, and then we've got insert the plasmid into the host bacterial cell. So in stage X, what I've got to do is to join the gene and the plasmid together. So they're asking for a suitable description of X. Insert the bacterial plasmid into the required gene. No, it's the other way around. Okay. Insert the bacterial plasmid into a source chromosome. No, not sure where that's coming from. Insert the required gene into the host bacterial cell. No, because it has to go into the plasmid and then into the cell. Insert required gene into bacterial plasmid. That we like. Okay. Question 7. The diagram shows an experiment which can be used to find the energy content of different foods. Each food was completely burned and the energy content was estimated by the rise in temperature of the water. And we have a little picture of a pretty straightforward one that we can do in the lab. Okay, there's lots of issues with it, but they're asking about the reliability of this experiment could be improved by. So reliability is whether you trust the answer that you're getting. It is always about, can you find some way of repeating? And they've got it just straight there. Repeat the experiment with each food several times. Okay, just to be absolutely clear, burning each food for the same length of time would not change the reliability of the experiment, but it might be something that you think is more correct in terms of the validity of it. Removing the thermometer to read it accurately would just mess everything up, because that means that you are likely to lose heat as you take it out. Repeat the experiment using a different food each time would obviously then be looking at different foods, not making it reliable. So definitely B. Okay, 8. The apparatus shown was used to investigate the rate of respiration in yeast at 20 degrees C. Which of the following changes would cause a decrease in the rate of respiration of the yeast? Okay, so I need to cut down the respiration level. So you know that temperature is involved in respiration, and if I cool this down, if I drop that temperature, that would decrease my rate of respiration. You know that you need a food source, so that 5% glucose solution, if I drop that down, that would decrease what's going on with the yeast. The rest of it has nothing to do with the rate of respiration of the yeast. The layer of oil is basically just stopping air from getting in, because we're looking at fermentation here. And the indicator solution is just showing what's going on, but it's not actually part of the yeast, kind of part of what's going on. Okay, so it's not about increase the thickness of the oil. Increasing the temperature would probably make it go faster. Decreasing the concentration, that would definitely impact on it. Decrease the volume of indicator's got nothing to do with it. So C is the answer for that one. Question 9, reflex arc. Okay, so we've got P, R and Q. Which one identifies which? So the nerve ending for P, so that's coming in the way, and then going round and then coming out to the muscle. I think that's probably the muscle is the only one that is absolutely crystal clear what you're looking at. So Q is the motor. Okay, the one in between is always the inter. So P is the sensory. So let's find the right one on that. So D. Okay, we've then got a diagram of your brain. Well, not your brain, just a brain. Which of the following links a letter to its correct structure and function? Okay, so up here we've just got G and H being given. This is the cerebrum, and H is the cerebellum. This bit here, they're not labelling, but that's the medulla. Okay, the cerebrum is all your thought, your memory, your reasoning, everything. Okay, so A is correct. Just to be clear, G is not the cerebellum, so that's why B is wrong. H is not the medulla, it is the cerebellum, but that means it controls coordination and balance. It's not about breathing and heart rate, that is the medulla. So A, definitely. 11 and 12 are to do with the following flow diagram relating to blood glucose regulation. You really need to know your stuff for this question. Okay, so we've got organ X releases a hormone Y which acts on organ Z and then promotes the conversion of glycogen to glucose. Okay, so what that is, just although it doesn't ask you all of this, but what this must be, if it's glycogen to glucose, then this hormone must be glucagon. Okay, organ X must be the pancreas, and organ Z would be your liver. So X is the pancreas, and the hormone is glucagon, hence D. Okay, because it's this bit here, the glycogen to glucose, that you need to have got for the glucagon, because help, my glucose is gone, kind of thing. Okay, question 12 says, specialised cells allow organ Z to respond to hormone Y. This is because the surface of the cells in organ Z have complementary, just got to know it, they have complementary receptors, okay, because they have to lock on. Question 13, an increase in which of the following factors would decrease the rate of transpiration in plants? Okay, so transpiration is basically water coming in at the roots, going up through the stem, getting up to the leaves, and then leaving. Okay, so anything which will increase the rate that you get movement of water away from the leaves, that's going to increase transpiration rate. But we're looking for a decrease in transpiration rate with an increase in the factor. Okay, if it gets windier, for wind speed, so the windier it is, the more transpiration you expect. So it's an increase. Humidity, that's making it kind of higher and higher water concentration on the outside of the plants. So increasing that is actually going to decrease that, because it's going to be less easy for the water to kind of come out. So that's that one. Increasing surface area would increase transpiration rate, and increasing temperature would increase transpiration rate again. So definitely B. 14. Which of the following statements about blood cells is false? So we're looking for an incorrect. White blood cells are part of the immune system. Absolutely agree. Phagocytes are a type of white blood cell. Yep. Red blood cells contain hemoglobin. Definitely. Phagocytes transport nutrients about the body. No, they are part of your immune system. So D. Which row in the table identifies how lymphocytes destroy pathogens? Okay, so lymphocytes are your other type of white blood cell that's part of the immune system. You've already got phagocytes mentioned up here, but now we're going to lymphocytes. Lymphocytes make antibodies, and the phagocytes do phagocytosis, so the lymphocytes do not. So it's A. Again, kind of definitely a wee niche bit of knowledge in that one that you needed to know for 1415. Question 16 is a key. The following key can be used to identify different components of blood. Use the key above to identify which of the diagrams represents a platelet. Okay, so you're having to work back the way here. Platelet. Diameter less than 0.005 millimeters. Well, actually, you've already got the answer. To take it back the rest of the way, it was number 2. So find the one that said go to 2, and the first one would have been nucleus absent. So these guys would have been out. This one doesn't have these two. We can't say nucleus in, so you would add C and D to go, and then, yeah, D. So you could get it in one. Question 17. Which of the following statements is true of villi? Okay, so we've got, these are our inside the gut, our kind of little finger-like projections, which have 2 points inside them for absorbing. So you've got blood capillaries, kind of all inside. Okay, they are taking in your amino acids from protein digestion and your sugars from carbohydrate digestion. They go straight into the blood capillary. And what you also have is inside the middle of it, you have a kind of central line with a different transport system. This is the lymphatic system, and that central point there is the lacteal, and the lacteal is taking in your glycerol and fatty acids from fat digestion. Okay, so what we're looking for in this one, which is true, blood capillaries absorb glycerol and fatty acids. No, they don't take in glycerol. Blood capillaries absorb glucose and fatty acids. They don't take in fatty acids. Lacteals absorb glycerol and fatty acids. Yes. And just to be clear, lacteals absorb glucose and amino acids. Definitely wrong, because that's just totally wrong. It's blood capillaries. Okay. Question 18. An ecosystem consists of abiotic factors plus A. So ecosystem is all living and non-living. So I need every single living thing there. So I'm going to need the community, not just a single population. And I need everywhere it lives. So I need its habitat as well. So D on that. Question 19. We've got a food web. Okay, a chemical was used to control the number of slugs. Which of the following could be a result of a large decrease in slug numbers? So we took out these guys. Okay, so I'm expecting my oak tree has got one less thing eating it, so it's going to be in a better state. So I'm not expecting a decrease in oak trees. Okay, the thrush has probably got a problem because it's now got less things to eat. Because it's got less things to eat, it will probably start eating more snails and more caterpillars, so they will go down. So I'm expecting a decrease in snails and a decrease in caterpillars, so C is correct. An increase in hawks would not happen because if we're saying anything, we're saying that the thrush numbers might go down a little bit. They might not, they might just stay the same because they've eaten more snails and caterpillars, but then the hawks would still just stay the same. Question 20, had to make this a little bit smaller so it would fit totally. Let's see if I can get it all on. Investigation into seed germination. Okay, so we've got 6 test tubes here. We've got different numbers of seeds. We've got different conditions for the cotton wool, whether it's dry or wet, and we've got different temperatures. For a valid conclusion to be drawn, which 2 tubes should be compared to show the effect of temperature on germination? Okay, so if I'm just looking for temperature, I need my seed number to stay the same, and I need the cotton wool to either be, to be the same as well, so either wet or dry. Right, so I'm looking at temperature. So if I look at the tube 1 has 10 seeds, so let's look at other ones that have got 10 seeds. The one next to it, tube 2, has got 10 seeds, but wet cotton wool, and that's dry and wet and change in temperature, so it's not 1 and 2. It's not 1 and 5 either, because that's the same temperature, and that's a change in the cotton wool. We were going 1 and 3 doesn't work because we've got the same temperature, same type of cotton wool, like they're both dry, and it's the number of seeds. 4 4 and 3, where we're going 20 seeds dry, and dry 5 to 20. So that might be in with a runner. And then we've got 5, we've got wet, that was our 10 seeds again. And 6 has got 20 wet and 20 degrees. Okay, so we've got various different ones in this. Let's check them on the actual answers. I think I might have been waffling a little bit there. Right, let's check 1 and 3. So 1 and 3, we have changed the number of seeds, and we haven't changed the temperature at all. So definitely not 1 and 3. 3 and 6, we've got the same number of seeds. We've got dry and wet cotton wool though. We have changed the temperature, but we've changed the cotton wool, so therefore we don't know if it's moisture or temperature, so not that. 2 and 5, we've got wet cotton wool, 10 seeds, 5 degrees and 20 degrees. So 2 and 5 works. And just to be clear, 4 and 6 don't work because we've got dry cotton wool, wet cotton wool, and the temperature is the same, so it's got nothing to do with that. Apologise for the slight waffle in that one. Question 21. Diagram represents a pyramid of energy. We've got X at the top. There is less energy at level X in the pyramid because, okay, so this is nothing to do with the number of organisms or the size of organisms, because that would be to do with pyramids of numbers. So get rid of A and B straight off. This is to do with energy, because it's a pyramid of energy. Basically, you should know that we lose energy at each level as we go up, which is why we end up with less at the top. We're expecting that you're only getting a 10% kind of bump each time that you go. So you're losing 90% of the energy at each level. That's a lot. Question 22. Mutations result in changes to genetic material. Which of the following is not true of mutations? Radiation can increase their rate. Definitely true. They always have a harmful effect. Now that's not true. They often do, but sometimes they're good for you, which is why evolution has occurred. You're the result of billions of years of wonderful mistakes. Okay, and just to be clear, genetic material is affected at random. That's your definition almost of a mutation. New alleles may be produced. That's the whole point of why we need them from a natural selection point of view. Okay, natural selection occurs when there are selection pressures. Which of the following could be a result of selection pressure? So once you've actually brought the pressure to bear. Okay, so what should happen is that we have whatever alleles are good for the organism, whichever ones give them a selective advantage, they will survive and reproduce. And there we go. Organisms with new alleles always have an advantage. That's not fair, given that we've just said that answer was incorrect for the one above. All alleles in a population increase in frequency. Well, if they all increase in frequency, well, that's actually impossible. You couldn't do that because of all alleles. And the same for all alleles decrease. There has to be changes. So it's not these two. Question 24. Pesticides sprayed onto crops can get into food chains. The following statements refer to stages in this process. So we've got pesticides absorbed by the plants, they build up in animals, and then the plants are eaten by animals. Okay, identify the order of steps that they could reach lethal levels. Well, this doesn't make sense the way it's been given. Okay, pesticides absorbed by plants, that would have to be your first one. Okay, then plants get eaten by animals, and finally they build up to the point where they start doing the animals harm. Okay, so first step has to be J. Second step has to be, we've said L. Oh, well, you're good then. D is fine. And third step has to be K. So D. And last one in the section. Which row in the table identifies biotic and abiotic factors that can affect a population? You're just going to go through and make sure you're happy. Okay, biotic must be living things. Grazing, predation, yes. Predation is good, temperature is not. pH and temperature both are not okay. pH is not, and grazing is. So actually you're already on this for A. But just to check, pH is abiotic, it's non-living, so is temperature. So A is correct, but to run through the rest, pH abiotic, grazing is biotic, grazing, predation are both, oh, I'm not putting crosses where I should. Grazing and predation are both biotic, sorry. Predation is biotic and temperature is abiotic. So the only one that has both is 25, is A, question 25. And that's that section.